All right, welcome everybody to another Xylella seminar. So today we have the pleasure of listening to the research of Kunle Olawole. He's an assistant professor in the Department of Microbiology and Plant Pathology at the University of California, Riverside in the US. His research focuses on plant-phage-bacterial interactions, particularly in Xylella fastidiosa and Pseudomonas syringae, with an emphasis on host range mechanisms, evolution dynamics, and sustainable phage-based disease management strategies for woody perennial crops. Prior to joining the UCR as a professor, he worked on projects related to olive quit decline syndrome and phage-based control strategies for Xylella. So today he's going to tell us about his research of comparative genomics of plant-associated Xylella fastidiosa subspecies multiplex. So thank you, Kunle, for giving this talk, and we're looking forward to learn more about your research.
All right, thank you very much, Leo. I appreciate that. Thank you for that introduction. Well, good morning, good afternoon, good evening, depending on what part of the world you are. So today I'm just going to be giving just a brief overview of one of the main projects that we have been working on in my lab, and this is based on the plum-associated Xylella fastidiosa subspecies multiplex. Just going to give an overview of it's going to be more of a kind of comparative genomic analysis and all of that. So, okay, so before we get going, just before discussing the genomics, I would like to briefly explain how this project originated. So this project actually began somewhat unexpectedly during my postdoctoral work in Dr. Carlos Gonzalez's lab at Texas A&M University at the Center for Phage Technology, where I was trying to develop bacterial isolation and phage-based approaches for olive quick decline syndrome. And this was actually with with some collaborators in Italy. And so one day, Kevin Ong, who at the time was the head of the extension department and also the director of the Texas Diagnostic Laboratories, he walked into my lab and then he explained that Texas had repeatedly detected Xylella in plum orchards through a USDA-funded National Stone Fruit Survey. But no one had been able to isolate this bacteria. And so that challenge eventually led us to develop modified media and also and recovery workflows that allowed us to be able to isolate multiple plum isolated strains, which later became the foundation for the comparative genomics work that I'll be discussing today. This phylogenetic network kind of shows that all specialization has been a major driver of Xylella fastidiosa evolution and resulting in the emergence of several genetically distinct subspecies. And these subspecies range from fastidiosa, which exclusively infect grapevine, to pauca, which infect olive, citrus, and coffee, to multiplex, which has a pretty broad host range, including plum, almond, peach, shade trees, and all of that, and also to morus, and also to syringae. So all specialization has shaped diversification across Xylella fastidiosa, producing the broader context for studying diversification within the subspecies multiplex. This subspecies multiplex is one of the most genetically diverse and broadly distributed lineages, infecting a wide range of woody perennial ores. If you look at these images here, let me see if I can use my laser point. Okay, so these images here illustrate the diversity of diseases that are caused by subspecies multiplex, including the plum leaf score, the almond leaf score, Chefoni peach, and the pecan, And all of these are Scotch symptoms. And so this subspecies has an exceptionally broad host range, with reports from approximately 250 woody plants, plant species, and it is also highly diverse genetically, with more than 40 sequence types described to date. So this combination kind of suggests extensive evolutionary diversification within the subspecies. Despite extensive studies that compare subspecies, much is less known about diversification within the subspecies multiplex. Previous genomic studies have primarily focused on differences between subspecies. Comparatively little attention has been given to variation within subspecies multiplex. And the evolutionary forces, particularly that shape diversification within this particular subspecies, remains unclear. To investigate diversification below the subspecies level, we focus on plum leaf scored as a natural model system. The plum-associated populations occur that I'm going to be talking about here occurs in both California and Texas, and also the system allows comparison of isolates that are exposed to distinct ecological environment. And also one of the things we notice, which I'm going to be talking about in a moment, is that we observe some phenotypic differences between the strains, which kind of suggests some underlying genomic differentiation. And looking at this figure here, this is a typical symptom of a leaf scored in agricultural plum. One of the first things that stood out to us was that the isolates that we're going to be talking about today were not only geographically distinct, but they also originated from very different host contexts. To be specific, the Texas isolates were recovered from commercial agricultural plums, and whereas the California isolates were likely associated with ornamental plum traits. So these were in Texas. This was when I visited the field, with Ong. When I was in Texas, I can actually see those symptoms. Those are it appears to be a very serious disease in Texas. You can see when I saw this picture, it kind of reminded me of the quick olive decline in Italy. So we're going to be talking a little bit more about this, particularly the kind of sequence types that are associated with the isolates that were recovered from Texas. So agricultural and ornamental hosts likely experience different selection pressure. And these contrasting systems provide a natural experiment for studying diversification. Okay, so to address some of these questions here, some of the questions as to what is probably driving some of the differences that we've probably seen in terms of agricultural versus ornamental, plums. We combine phenotypic assays with comparative genomics. First, we isolate and purify Xylella fastidiosa from symptomatic plum tissues. They were collected in Texas and California, and also we did some phenotypic characterization ranging from pathogenicity to biofilm and also to planktonic growth. And then we send those strains also for sequencing and also for a form of a comparative genomics analysis, and also lastly we perform some accessory genome analysis. On the strains. The first question we asked was whether California and Texas populations differed, phenotypically. And generally speaking, what we found was that quantifying disease using the area under the disease progress curve values, we noticed that generally speaking, the Texas isolates generally produce higher disease severity than the California isolates. I mean, these are the number of isolates that we had at the time when we conducted this study, and we are currently expanding the California isolates, and by now we have a lot more California isolates. But at the time when we performed the study, we had all of these Texas isolates and just 3 isolates from California. And this was actually because these strains actually really they are quite challenging to isolate, and that's why we probably don't have a lot of those collections out there in public. So the California isolates consistently exhibited lower virulence. And the red violins here, this represent the Texas isolates, and then the blue represent the California isolates. And if you look at it, the Texas isolates occupy the upper portion of the disease severity distribution, and what the California isolates cluster near the lower end of the distribution. And the phosphate buffer saline was used as a control. We should know this. We next asked whether the Texas and California isolates differ in growth and biofilm formation. One of the things we found was that the Texas isolates exhibited reduced planktonic growth. So generally speaking, if you look at it, this is just generally speaking like a kind of pattern that we observe among just a subset of the Texas strains that we looked at in this particular study. And also, if you look at the California isolates, they appear to be a little bit more they show faster planktonic growth. However, one of the things we quickly noticed was that the Texas isolates produced substantially more biofilms, even though their growth rate was lower. The biofilm was, was a little bit higher. There's a pattern of some of those strains of being higher. But if you look at the California strains, which exhibited higher planktonic growth, they exhibited much lower biofilm formation. So this inverse relationship between growth and biofilm formation, kind of suggests an alternative ecological strategies. So to place our isolates into a broader evolutionary context, we compare them against publicly available Xylella fastidiosa subspecies multiplex genomes. So we sequenced 10 strains that we had at the time, 7 from Texas and 3 from California, and we integrated those with 40 publicly available multiplex genomes that represent diverse hosts and sequence types. Now we perform some core genome, ancestry genome, and population structure analysis, and then we use these comparisons to determine whether the plum-associated isolates fit within the global diversity of the subspecies multiplex. This is the core genome. The core genome phylogeny of these strains reveals that the plum association has evolved in multiple evolutionary lineages, rather than just a single plum specialized lineage. Now, if we take a look at this graph here, the red strains were the ones that we sequenced, that we isolated and sequenced in this study, and the blue strains were additional plum isolates that were retrieved as part of the foray that we retrieved from public genome database. And if you look at it here, you will see that instead of forming a single lineage, we can actually see multiple lineages that are driven or rather associated with the sequence types. And one of the things that was became very clear here was that the ST26 and also the ST34 represented the dominant plum-associated lineages in our collection. Additional plum isolates occurred, which include the ST6, the ST10, and also the ST41. So which kind of indicates multiple independent evolutionary backgrounds for this plum infecting isolate. The ancestry genome also revealed that the plum-associated isolates can share similar gene repertoire across genomic regions and also sequence types. The ST34 isolates, of course, they also formed. The ST34 isolates are consistently also they formed a distinct ancestry genome cluster, regardless of whether they originated from Texas or not. And one of the things I forgot to mention was that within the ST34, we had strains from Texas and also strains from California. But within the ST26, those were exclusively strains from Texas, which were the ones that were recovered from agricultural plum trees. The Texas ST26 isolates also clustered with the plum-associated ST10 10 isolates from Georgia, and also an ST26 isolate from Brazil, which indicates a shared ancestry gene repertoire. So this clustering suggests that the plum associated strains from different regions may share lineage associated ancestry genes, despite geographic separation. So overall, the ancestry genome appears to be a reflection of the shared evolutionary history and also the gene content among among the identified lineages, rather than just the geography alone. The ancestry genome further supports the existence of multiple plum-associated lineages with distinct gene. Just to kind of buttress the previous slide talking about the ancestry. Talk about the ancestry phylogeny. The ST34 isolates are also shared. You can see that it shared a highly conserved ancestry gene pool. And this was regardless of whether they originated from Texas or California. And also, if you look at the ST26, so this isolate possess a distinct ancestry gene repertoire, which kind of separates them. There's an offset plot, and these lines you are seeing there, they're just talking about the genes that are shared by different strains. And so if you look at the ST26, they possess a distinct set of ancestry gene repertoire that is separate from the ST34. And also the Texas ST26 isolates also grouped with the plum-associated ST10 from Georgia, and also ST26 from Brazil, which also indicates a shared ancestry gene. You can see that pool of genes that are being shared by this group. So to determine whether our initial observations were representative of the broader plum-associated population, we incorporated additional plum-associated genomes from this time around from California. Remember, the focus was meant to be on Texas and California strains. So if you look at it on the left here, this was with 15 isolates, and we were able to retrieve additional green. These isolates that are in green. This was like additional 8 that we were able to add to this, and this included 7 from California and 1 from Texas. And one of the things that we quickly noticed was that the addition of these new strains introduce new sets of sequence types. So that's from California. Initially, we had always thought we had thought that or rather based on what we found initially, all of our California strains were restricted to the ST34. But now with additional California strains, we're able to identify strains that clustered with ST6, and then another one that clustered with ST23/45, and then 4 other strains that cluster with ST39. And of course, the ST26 and the ST34 consistently remain, separate lineages. And also the additional texture strain that we added also clustered with the ST26. And again, remember this ST26 lineage. These were isolated from agricultural plants, and all of the California strains were isolated from ornamental plant. And also, let's ask whether the expanded sequence type diversity observed in the core genome was also reflected in the accessory genome composition. And so what we found was that the accessory genome relationships closely mirror the sequence type assignment with the newly added, ST6, ST23, 45, and also the ST39. So the gene content variation varies. It provides additional resolution beyond the core genome phylogeny. Because functional validation, often requires access to live isolates, we decide to focus. Excuse me. We decide to focus subsequent analysis on the California and the Texas lineages that are available in our collection, because the goal is to be able to support the drivers of some of these differences that we've seen so far. And so if you look at this here, these were all of the Texas and the California strains. So right now I'm trying to switch colors, trying to use the political color here. Excuse me. So the red, those are the Texas strains, and the blues, those are the California strains. And as you can actually see here, that our collection contains representatives ST26 and ST24, which consistently remain the main ST groups in this particular study. So these accessible lineages provide a framework for linking phenotype, genotype, and also future functional validation experiment. Despite representing different evolutionary lineages, the major plum-associated sequences, sequence types exhibited remarkably similar overall genome organization. And if you look at it here, all of the STs, all of the sequence types contain similar proportions of genes. As we found in this range from virulence to mobile to carbohydrate regulation, and all of these are different functional groups. Of course, the largest fraction of these genes in every lineage consisted of the hypothetical and poorly characterized proteins, as you would expect. So therefore, diversification among the plum-associated lineages likely reflect differences in the specific lineage-enriched genes, rather than just the wholesale changes in the genome content. So this led us to kind of ask which specific genes distinguish one sequence type from another. Although the overall genome architecture was conserved, each lineage possessed a relatively small set of lineage-enriched genes that distinguish it from the others. And now looking at this here, we're going to see that most lineage-enriched genes were associated primarily with hypothetical proteins, like we saw in the previous slide, and also phage or prophage, phage or prophage enriched genes. And also, if you look at the ST, however, the ST26 contained the largest number of enriched genes associated with that is associated with multiple element, mobile element genes. You can see that the ST26 and also the ST26 also here. So if you look at it, the ST26 also contains some genes, some functional groups that were kind of restricted to that ST26. Like if you look at the transport and secretion, also virulence, addition, surface, these were consistently present in the ST26, and of course we also found that in the ST45, and we found a lot of differences in terms of the some of those genes being less enriched in the ST34 and also the ST39, and also ST6 also was missing some carbohydrate and APS metabolism gene. So this kind of tells us that the lineage differentiation is concentrated in a relatively small set of genes, with the ST26 kind of showing the strongest enrichment for mobile elements and also host putative host interaction functions. The ST34 provided a natural experiment to evaluate the effect of geography while controlling for lineage. Now, if you look at it, one of the things we found, like I mentioned earlier, for the ST34, that was just so unique about them was that this particular sequence type comprises Texas and California isolates, and one of the things we found was that these isolates remain genetically similar despite their geographic separation. However, one of the things we found, like I mentioned earlier, was that the Texas ST34 isolates exhibited higher virulence than the California ST34, and also the Texas isolates produce more biofilm, and they grow less, more biofilm than the California isolates, and also they don't grow as much. They are slower growing compared to the California isolate. So these phenotypic differences emerge despite the minimal genomic differentiation that we observed between these strains. So that led us to wanting to ask additional questions as to are there differences, putative differences probably at the ancestry genome level that could be driving these phenotypic differences. So we next tested whether geography, contributes substantially to the genomic differentiation between the dominant plum-associated lineage, which was ST34 and the ST26. But before then, we wanted to see what if there's a difference between these between the ST34 Texas, between the California ST34 and the Texas ST34. Now, one of this we found here was that about 2,322 ancestry gene clusters were shared between the California and the Texas, and only 4 and 5 genes were unique to California ST34, and 5 were unique to the Texas ST34. So more than 95% of the ancestry genome was shared, basically. And so this results indicates that the geography contributes very little, okay, to the ancestry genome variation within the ST34. So the next thing I wanted to find out was, okay, well, why not let's go into the core genome and see if we probably maybe what is driving these observed differences is probably there are probably genetic variations in terms of single nucleotide polymorphisms. So we did this for the core genome of the California ST34 and the Texas ST34. And what we found was that it was just 6 polymorphic core genome sites that were identified among the ST34 isolates, and most SNPs were shared across California and Texas isolates. So none of the SNPs showed a fixed California versus Texas differentiations. In other words, we didn't see a particular SNP that were probably conserved within the Texas strains, and some that were conserved within the California strains. So these results kind of provide little evidence for geographic structuring at the core genome level. So to just kind of wrap this up here, what we found was that the largest component of the variation is the shared ancestry genome within the ST34, and also that region-specific genes represent just a really tiny fraction of the total genomic content. So So geographic origin alone explains very little of the genomic variation that we observed within the ST34. And the biological differences observed between the California and the Texas isolate likely arise from probably higher other lineage effects, including mobile genetic elements, regulatory variation, or other mechanisms that are not captured by the simple presence, absence, or SNP analysis that were carried out within this ST34. So having established that the geography contributes little variation within the ST34, we next ask whether the major genomic differences in the plum-associated population are associated with sequence types. Like I mentioned earlier, the ST26 and ST34, they form the major phylogenetic groups in the data set. And one interesting thing about these two groups is that both sequence types are recovered. They have they have strains that are recovered from agricultural plum strains, with the ST26 being exclusively from agricultural plum strains, plum hosts, and then the ST34 being a mix of both the ornamental and the agricultural plum. So the minimal geographic differentiation observed within the ST34 suggests that broader diversification may probably occur at the sequence type level. So these observations justify direct comparison between the ST26 and the ST34 to identify the major genomic features that are associated with this diversification. So if the sequence type is the primary axis of diversification, for example, we would expect ST26 and ST34 to probably share a common genomic background while retaining lineage-specific gene repertoire. But one of the things we found was that the ST26 and the ST34, they share a large core gene set of about 1946 genes. And that's why this shared backbone, about 246 genes were enriched within the ST34, and a little bit more than that were enriched within the ST26. So therefore, hundreds of these ancestry genes, we believe, distinguish the two dominant plum-associated lineages. And this lineage-enriched genes represent strong candidates. And so that's the long-term goal of this, and that's what I'm going to get there in a moment. Candidates for exploring differences in virulence, biofilm formation, and also ecological adaptation. To better understand the biological basis of the ST26 and the ST34 differentiation, what we did was to examine the functions that are associated with lineage-enriched genes. And one of the things we found here was that most of the sequence type enriched specific genes were associated with hypothetical and also with hypothetical or poorly characterized proteins. A large fraction of the lineage-enriched genes were linked to prophages. They were linked to prophages or probably phage-related genes. And the ST26 contained more enriched genes. That was one of the things we found that was so, that became very obvious. They contain more enriched genes overall, including genes involved in toxin, antitoxin systems, transport secretion, virulence addition surface. And this is some of the genes that were not reported in the ST34, or they were less enriched within the ST34. So these patterns kind of suggest that mobile DNA and uncharacterized genes are the major contributors to lineage diversification. But trying to validate this hypothesis is completely outside of the scope of this talk today. So to determine whether the ST26 and ST34 are differentiated by fewer genes or by broader patterns of gene content, what we did was to examine the distribution of the ancestry genes across the two lineages. And one of the things we found was that many of the ancestry genes, many ancestry genes showed near lineage-specific presence and presence-absence patterns. Now, if you look at the SC26, for example, the SC26 contains a larger number of highly enriched genes than the ST34. This is just kind of providing additional layer of evidence to support the previous slide. And then most of the lineage associated genes were either present in nearly all members of one lineage and absent from the other. So this results indicate that the ST26 and ST34, they are genomically distinct lineages, despite sharing a large core, genome. So this lineage differentiation is driven from what we've seen so far by numerous strongly lineage associated ancestral genes rather than just a number of genomic changes. Okay, that being said, our findings raise an important question. And this question is that should ST26 be viewed as an emerging agricultural risk lineage within the Xylella fastidiosa subspecies multiplex? And this is actually based on the fact that the ST26 were associated with highest virulence and biofilm phenotypes that were observed in this study, and also that the ST26 possess a distinct ancestral genome that are enriched in mobile elements, EPS associated, and also host interaction functions. And also recently, the ST26 has now been reported from plum in USA and Brazil, and also more recently in almond and sherry in Italy. This paper was published just last month. That's less than a month ago, and which kind of indicates a broader agricultural footprint than previously appreciated. So this observation suggests that the ST26 may represent a globally distributed agriculturally important lineage which cannot deserve increased surveillance and probably functional investigation. So that's going to be the major take-home point from this slide, and then from this talk today. So sequence type, just kind of wrap everything up. What we found was that the sequence type rather than geography is the primary axis of diversification in the plum-associated Xylella fastidiosa subspecies multiplex. With the key point here being that the ST26 is associated with higher virulence, stronger biofilm formation, and a distinct ancestry gene repertoire that kind of suggests a potentially important agricultural risk lineage. And also for the ST34, it's broadly distributed across California and Texas, and yet it exhibits minimal genomic differentiation across regions, making it a useful model for studying local adaptation. And so most of the genomic variation is explained by lineage-specific genes and also mobile genetic elements that are not geographic or that are not and rather not geographic origin. So future work is going to focus on functional validation of some of these lineage enriched genes, and also plasmids and prophages. We perform some of those analyses, but of course we are not presenting them. That's going to be too much of a study to include in a single presentation. So together, this finding suggests that evolutionary lineage and then not geography is the major determinant of diversification in plum-associated Xylella fastidiosa subspecies multiplex. And with that, I want to say a big thank you to members of the Olawole lab, the current members, Kwong Ryan, Chioni, Yisin, Jackie, Jonathan, Dominic, Lisbeth, and Suraj. It's important that I mention their names. I know some of them are connected to this talk today, and also some of our previous members, and also our collaborators. I just want to say also thanks to our collaborators, Dr. Borbank, Dr. Roper, Dr. Uribe from Dr. Burbank and Dr. Roper, they've been very supportive in giving us, additional strengths to kind of work with. They've been working on Xylella for decades, and we really appreciate the opportunity to be able to connect with them. And Uribe and Brennan, they've been committed to sending us, consistently sending us infected plum materials, all the way from Texas to California, and Brennan all the way from Georgia to California, because the goal of this is to be able to hopefully get some isolates as well from Georgia and be able to see how those also map to our current phylogeny, and also the current phylogeny of the plum-associated Xylella. And also to our funding agencies, I just want to say thank you very much. I think with that, I'm just going to take questions and also comment. So let me just mention that this work is currently under review, and so we really appreciate any thoughts or a comment that could probably help getting this work published eventually. Thank you very much, and I'll take questions or comments.
Thank you, Conde, for a great presentation. So now again, we open for questions. You can either talk or write it in the chat.